Hello everyone. Let us understand the mechanism of oxidative phosphorylation. The mechanism is explained by a theory called Chemiosmotic Theory, which is proposed by Peter Mischer. So this oxidative phosphorylation occurs in the inner mitochondrial membrane where the energy released from electron transport from one complex to another is converted into ATP, which is universal energy currency of the cell. So let us understand the mechanism of oxidative phosphorylation. Okay, imagine this is a mitochondria where outer mitochondrial membrane and this is inner mitochondrial membrane. The space between outer and inner mitochondrial membrane is called intermembranous space, which is very important to understand the chemiosmotic theory. The inner space is called mitochondrial matrix where we know beta oxidation citric acid cycle takes place. There are different complexes which are located in the inner mitochondrial membrane. They are called complex 1, complex 2, complex 3 and complex 4. In addition to these fixed complexes in the inner mitochondrial membrane, we have mobile electron transport carrier, which is ubiquinone or coenzyme Q and cytochrome C. So as a result of metabolism, whether it could be carbohydrate metabolism or lipid metabolism, they generate reducing equivalents or electron-rich coenzymes. They are called NADH plus H plus and FADH₂. In citric acid cycle and beta oxidation, they are generated within mitochondrial matrix, whereas in glycolysis, they are generated in cytoplasm and transported across to mitochondrial matrix. Now, NADH plus H plus donates its electron to complex 1 and these electrons are transported from one complex to another complex. So that means coenzyme Q accept electron from complex 1 and hand over to complex 3. From the complex 3, cytochrome C accept electrons from the complex 3 to the complex 4. Similarly, when FADH2 converted to FAD, it will donate electrons to complex 2, not complex 1. This is the point to remember in order to understand energy yield or ATP yield. Okay, so now whether through FADH2 or NADH plus H plus, we have electrons in the complex 4 which eventually hand over to oxygen atom along with the two protons to produce one water molecule. Okay, so the whatever electrons transported across various complexes from the reducing equivalents which are generated as a result of metabolism, the final acceptor of electron is oxygen. So that is why it is called respiratory chain. Now these transfer of electrons across different complexes, it involves various oxidation reduction reactions. That is why it is called oxidative. Now during this process, that means transfer of electrons from one complex to another complex, at the complex 1, 4 protons are pumped from mitochondrial matrix into intermembranous space. Okay, clearly you can see 4 protons are transferred from the matrix to intermembranous space. So when this occurs, when NADH plus H plus donates 2 electrons to complex 1 and complex 1 the electrons are handed over to complex 3. During this process, 4 protons are pushed to intermembranous space from the mitochondrial matrix. Similarly, when complex 3 handovers electron to complex 4 through cytochrome C, there is 4 protons are pushed from mitochondrial matrix to intermembranous space. Please note that there is no pushing of protons from the matrix through the complex 2 to the intermembranous space. Only complex 1, complex 3 and also in complex 4, 2 protons are pushed to intermembranous space. Please remember, when electrons are transported from one complex to another complex, protons are pushed across complex 1, complex 3 and complex 4. You also remember 4 protons are pushed to intermembranous space through complex 1 and complex 3, whereas only 2 protons are pushed to intermembranous space from the mitochondrial matrix. Important point to note, there is no proton pushing through complex 2. Through the coordinated action of complex 1, complex 3 and complex 4, protons started accumulating in the intermembranous space, creating both electrical and chemical gradient. Let me explain what is that. Okay. You can see more positive charge in the intermembranous space, whereas more negative charge in the mitochondrial matrix side of inner mitochondrial membrane. That is more positive outside and negative inside across inner mitochondrial membrane, creating electric gradient. So this is called electric gradient developed across the inner mitochondrial membrane. Also, there is a chemical gradient because there is less pH in the intermembranous space, more pH towards the matrix side. So this is creating chemical gradient. So together we can call it as electrochemical gradient as a result of transfer of electrons across various complexes and pushing protons to the intermembranous space. So together this electrochemical gradient, there is a force generated across inner mitochondrial membrane, especially in the intermembranous space. This is called proton-motive force. That is nothing but accumulation of protons in the intermembranous space. Now these protons should come back to mitochondrial matrix in order to make electrochemical balance or electrical balance. There is another complex called complex 5 or ATP synthase, which is also present in the inner mitochondrial membrane. Now the protons move back into matrix through ATP synthase. Proton moves back into matrix through ATP synthase, which has got FO portion which is membranous domain and F1 portion which is matrix domain. When protons moving through these proton channels or H channels of FO domain, there is F1 has got enzymatic activity. Again, there are alpha, beta, gamma, delta units or subunits which makes ATP or phosphorylation. So ADP and inorganic phosphate will give ATP. Please note that the FO portion has got hydrogen channel or proton channel and the C ring. So when protons moving through here, there is movement and there is conformational change in the F1 subunit or domain where it will produce ATP. So this is called phosphorylation. Here various oxidative reactions are taking place when electrons are transported from one complex to another. So together we can call it as oxidative phosphorylation. So as the proton flow through the FO channel, the rotor mechanism within F1 begins to turn, catalyzing phosphorylation of ADP and inorganic phosphate to form ATP. So this coupling of oxidation to phosphorylation is the hallmark of oxidative phosphorylation. So this is actually chemiosmotic theory or the mechanism of oxidative phosphorylation. So let us understand how much ATP is generated per NADH and per FADH. For this you need to remember NADH always pushes protons through complex 1 or it handovers electron to complex 1. Whereas FADH bypasses complex 1, only it handovers electrons to complex 2 then to complex 3. So with this background we can see the energy yield or ATP yield. So each NADH molecule yields approximately 2.5 ATP. Why? Because it pushes 4 protons to intermembranous space from the mitochondrial matrix through complex 1 and complex 3 also 4 protons and complex 4 2 protons. Okay. So they say that approximately when 4 protons are pushed, 4 protons when it come back to mitochondrial matrix, it will produce 1 ATP. So here 1 ATP, here 1 ATP and here half ATP. That is why NADH plus H plus give 2.5 ATP, whereas FADH2, it is handing over electrons to complex 2, so there is no complex 1 involved here. That means only 4 protons through complex 3 and 2 protons through complex 4. That is why FADH2 give only 1.5 ATP, whereas NADH plus H plus gives 2.5 ATP. Finally, you should know electrons move through complex 1 to complex 4, driving proton pumping and generating proton-motive force. So the stored energy then powers the ATP synthase, resulting in the formation of ATP. So oxidative phosphorylation stands as a central mechanism linking the oxidation of nutrients to the synthesis of ATP so that we can survive, we can do all the actions of life. Thanks for watching.